Alright guys, welcome back to Underground Science, and in this video we'll be going over another type of neuroplasticity in our brain called structural depression. Alright, so let's go and write that down, is that we said it's a type of neuroplasticity. Alright, and so if you already watched my structural potentiation video, it's going to be pretty much the opposite of that. Alright, so if you remember from the last video, right, from the structural potentiation video, we said that as we start to use this particular neuronal pathway more and more, right, we see that in our presynaptic neuron, we start to produce more axon terminals and more synapses, all right, on our postsynaptic neurons' dendrites. And so what will happen in structural depression is that as you start to use this particular neuronal pathway less and less over a long period of time, what we're going to see is that you're going to start to lose these synapses that you originally built. All right, so let's go and write that down first, what we just said previously concerning time. All right, so if we're talking about not synaptic, but if we're talking about structural depression, this is also known to occur over a long period of time. All right, so this is also known to occur over a long period of time. All right. So that's good to know. And so again, if you stop using this particular neuronal pathway over a long period of time, as we decrease the number of action potentials fired down this neuronal pathway, what we're going to see is that instead of having all these synapses, I should say, right, all of these, and this huge one right here, we're going to start to see a decrease in that. So instead of that, what we're going to see is this. So let's just leave the dendrites out just for the sake of time. And we're going to draw our axon and our axon terminal, right? So what we're going to see is that we're just going to have maybe, I don't know, one synapse right here instead of all those on the postsynaptic neurons dendrites, right? So we're going to have dendrites here, and we're only going to see one synapse, or one or two synapses, but the overall goal to understand here is that over time, as we fire less and less action potentials, over a long period of time, due to structural depression, we'll start to see one type of structural depression is that we'll start to lose our many axon terminals and synapses that we had here. All right, so to be more specific, we're going to start to lose the synapses, right, in particular, because these are the synapses of a neuron, right? Like this humongous one is a synapse, right? So this is the axon terminal, and then we have our synapse, but we build more synapses in structural potentiation over a long period of time, but in structural depression, when we talk about using this neuronal pathway less and less, which means we're not going to be firing many action potentials down this axon or down this neuronal pathway in general, we start to see structural depression, right, where we start to lose our number of synapses that we originally had. So that's one type of structural depression. So let's go and write that down. So we lose the number of synapses, right? We lose the number of synapses for the presynaptic neuron, right? So this is going to be for the presynaptic neuron. Presynaptic neuron. And then another type of structural depression is that, remember previously we said that in our structural potentiation videos that we said that we start to build more and more dendrites on the postsynaptic neuron over a long period of time as we use a specific neuronal pathway over and over again, fire more and more action potentials, right? So we start to build more dendrites in structural potentiation. However, in structural depression, as we can see, is that we start to lose some of those dendrites, right, that we originally built. So now we start to have less and less dendrites, all right? And what this is going to do is that we're going to have less dendrites, means less spaces to receive the signals from the presynaptic neuron. So we're going to pretty much slow up the postsynaptic neuron, right? So we're going to reach the action potential much slower rather than if we had a bunch of synapses right here and a bunch of neurotransmitters and a bunch of dendrites, we're going to be much less sensitive when we lose our dendrites. All right, so instead we'll only have a few dendrites like this. And so that's another type of structural depression, is that we start to lose dendrites over a long period of time. We start to lose dendrites on the postsynaptic neuron. All right, so on the postsynaptic neuron. All right, so that's the two types that I wanted to get to in this video, is that we have either we lose the number of synapses on the presynaptic neuron in structural depression over a long period of time, right? Or we lose the number of dendrites on the postsynaptic neuron over a long period of time. So this right here, so this is a good comparison point, is that this neuronal pathway represents our structural potentiation, and this neuronal pathway right here represents structural depression. All right, so that's that, right? So again, if we lose a number of synapses here, we're not going to have many neurotransmitters either, right? Because for every synapse, we release a certain amount of neurotransmitters. And so that'll decrease the chances of us having enough temporal and spatial summation due to our graded potentials. It'll decrease the chances of reaching threshold potential in the postsynaptic neuron's axon hillock. Okay, so overall, it's going to slow the action of this neuronal pathway down due to structural depression. All right, and so that's pretty much it for the two types of structural depression that I wanted to get through. And also, one thing to know is that in structural depression, what could happen is that if we stop using this neural pathway at overall, in general, we just straight up dip, right? And we stop producing this stop using this neuronal pathway, so which means we stop firing action potentials like 0, right? Then what that means is that we're going to undergo something in the brain called synaptic pruning. Okay, so if we stop transmitting action potentials at all, right, all the way, then, I mean, think about it in your muscles, right? If you just stop lifting weights, and over a long period of time, you'll start to see atrophy because your body doesn't need that muscle anymore because you're not using it, right? So it starts to use up that muscle tissue. A similar situation can be seen in the brain when the brain undergoes synaptic pruning. If you don't fire any action potentials down a neuronal pathway or a specific neuronal cell or a neuron over time, or over a long period of time, we can see something called synaptic pruning, right? We can see something called synaptic pruning, which is pretty much when the neurons that aren't really getting used at all can start to die, all right? So the neurons that aren't getting used can start to die, and because we wouldn't want extra neurons, right, if they're no use, right? So if we're not using those neurons, right, if we're not firing any action potentials down those neurons, we can undergo synaptic pruning on our brain, where our neurons can start to die, all right, along with the structural depression that occurs. Okay, so just like our muscles too, right? I mean, why would your body want to keep that extra muscle tissue if you're not going to use it? So I'm sure you've noticed that too if you've been going to the gym getting those gains, right? And then after a few weeks or even months, you notice that, oh no, you've had some atrophy of your muscle tissue. That's because you haven't been stimulating those lower motor neurons and actually breaking down the muscle tissue, so it doesn't have any reason to grow. So it's gonna get used up, for other metabolic processes in your body. Like you can use muscle tissue, the protein, amino acids to make sugar, or something like that. All right. So that's pretty much what I wanted to get through in this video. I really hope this video made sense to you guys. I hope to see you guys in my future videos. We'll talk about more cool topics about biochem, physics, and chem. Don't forget to hit that notification bell, like, and subscribe. See you guys later, and stay safe.